Okay, thank you again for the invitation and the presentation. So here I plan to show you my work in the last few years that is related to the analysis of global plankton biogeography. And the idea is behind these different pieces of work is to show you how the new technologies of omics and high throughput imaging can be integrated towards this or other type of analysis. So let me. So to provide some context, all this work, uh, all the work that I will show you consists of the mining of the dataset generated by the Tara Ocean expeditions. These expeditions were carried out for uh, taking seawater samples from the main ocean regions, including the Mediterranean Sea, Indian Sea, South Atlantic, South and North Pacific, North Atlantic, and finally the Arctic, as well as part of the Southern Ocean. One important thing about the sampling process is that it was used a serial filtration system that in order to filter seawater at different size so that each size fraction is enriched in a certain plankton size. In particular, they obtain six different size fractions from from viruses to then bacterioplankton and then large eukaryotes as well as symbiosis and colonial forming organisms. And these plankton samples were then analyzed in order to generate both molecular data as well as high throughput imaging data. The molecular data included DNA and RNA sequences, and the different type of high throughput images, including high throughput confocal microscopy, uh, optical microscopy, as well as other technologies. Um, the idea here is to use both molecular and imaging because they complement the information. With molecular data, we can detect very rare species, as well as those species that we don't know how they look, or that they are very different, that they have simple morphologies, so they are very difficult to classify according to the morphology. Whereas with high throughput with different type of imaging technologies, we are able to detect absolute quantification of the organisms and also biomass, or let's say estimate the biovolume, which is a proxy of biomass, which is very important from geochemical point of view. And also lifestyles, if they are symbionts, free living, colonial forming. So the use of the integration of these different data sets, it's very can generate new piece of information. And so I will give you three examples of how these integrative approaches were applied. First of all, I will start with nitrogen fixers in a project that we did together with Rachel a few years ago. Then a new project in which we analyzed the global phytoplankton community. And finally, a project that we very recently finished that consists in the specific analysis of diatoms. So here, starting with the analysis of nitrogen fixers. So to provide some context, most of the ocean is nitrogen limited, and because all organisms needs nitrogen for their as elements forming DNA, and proteins. So the input of new nitrogen to the ecosystem is carried out by a reaction, by a let's say biological reaction that is called nitrogen fixation, that consists of the fixation of atmospheric nitrogen into bioavailable nitrogen in the form of ammonium. This reaction is catalyzed by a complex called nitrogenase, and this complex is only present in a few prokaryotic lineages. And these prokaryotic lineages are called nitrogen fixers, and they vary. They are very, let's say, they cover a wide range of different lifestyles, from photosynthetic to heterotrophic, from free living to colonial forming organisms to symbionts. For example, here there is an example of some nitrogen fixer symbionts living inside a diatom. And also in terms of symbiosis, there are some nitrogen fixers in which the integration with the host is attached to the surface, and they can be free living at some point. Others that live, let's say, inside the cells. There are also some cases of integration that are now more recently considered to be already an organelle. So that's why, in terms of let's say, analysis is very useful to analyze them using not only the genetic information but also their morphology. For this project, the main questions that we wanted to resolve were who are the main nitrogen fixers in the ocean, where are they found, which are their different lifestyles, and are there ocean regions in which that consist of nitrogen, let's say, hotspots of nitrogen fixers due to the nitrogen limitation. For that, at that moment, we decided to analyze both metagenomes. I mean, the DNA, the samples generated from the DNA, the sequences of the whole DNA of the sample, as well as imaging data, in particular high throughput confocal microscopy. And the analysis started with the analysis of the metagenome, because this is something that we have already a huge amount of tools, bioinformatic tools, to analyze metagenomes, and this is much more easy nowadays. Not only due to the bioinformatic tools, but also because we have big reference databases in which we can compare the DNA from the sample with DNA from organisms that we know. And for this analysis, we use two different gene databases. One for the gene nifage, which is a subunit of the nitrogenase and is a proxy of nitrogen fixers, and other database for the gene REK, which is a proxy of bacteria. And then we align the metagenomic reads against these databases in order to consider the relative ratio of metagenomic reads between nifage and REK as a proxy of the relative abundance of nitrogen fixers in the bacterioplankton community. So as I mentioned, we have the, let's say, metagenomes generated from obtained from different size fractions due to the use of a serial filtration system. Some of the patterns that we obtain when comparing the different size in using metagenomes is that there is an increase in the relative abundance of nitrogen fixers when going from the smaller to the largest size fraction. This is there is also a shift in the taxonomy with the size from proteobacteria. I mean, the smallest size fraction more enriched in proteobacteria, whereas the larger size fraction more enriched in cyanobacteria. And is a small nitrogen, and also we found a patterns in terms of let's say environmental distribution. Smaller nitrogen fixers detected in the smaller size fractions are more widespread in terms of nitrate concentration and in terms of temperature, whereas those abundant in the larger size fractions are more constrained to temperate waters with limited in nitrogen. And so all these patterns are associated with the let's say the different lifestyles in the different size fractions as I previously mentioned. So in the smaller size fractions, we expect to detect those nitrogen fixers that are free living, whereas in the larger size fractions, those that are symbiont or colonial forming. And well, all these patterns of environmental constraints in terms of temperature, for example, between the size fractions are very clearly illustrated when plotting the biogeography, in which we see that for the smaller nitrogen fixers, they are widespread in terms of latitudinal gradient, but with the larger nitrogen fixers, they are more constrained to tropical and subtropical regions. And this is important, very useful information in the context of climate change and the tropicalization of the oceans. So again, this slide basically illustrates the same results. But here, so far, I only mentioned let's say the traditional 5 size fractions. But there is another size fraction that consists of the seawater that passed through a pore of pores of size 0.2 microns, and this size fraction is considered to be enriched in phages in bacterial viruses, and so it's usually analyzed for targeting these viruses. But that moment we also decided to analyze these size fractions, and we detected a high abundance of nitrogen fixers that were not detected in the other size fractions, and this was surprising, but it was even more surprising to see that these what we call ultra small nitrogen fixers were especially located in the Arctic, where the rest of the nitrogen fixers whereas the nitrogen fixers from the other size fractions were not very low abundant in that region. But so far, those were results from the metagenome, from DNA. The most challenging part was to analyze the imaging data and to integrate that with the analysis of DNA. From the imaging data, the problem is that we need, there are not, let's say, available big reference databases, or we have to, let's say, we have to build the reference databases according to the imaging technology that we are using. So the way of, let's say, the workflow for annotating nitrogen fixture images consists of um, gather example images, what is called a training set of images, generate a machine learning model that learns from that example images in order to predict new images in the whole data in the whole data set of the whole data set that consisted of 2 million images. Nitrogen fixtures are very rare, with the except of some examples of blooming, but in general they tend to be rare. So it was very challenging first to obtain these example images. So the workflow of the decision here consisted in come back to the metagenomes in order to select a few metagenomic samples in which we detected a high abundance of NIFH. Let's say three samples with high NIFH values, and then go to the parallel imaging samples and manually select nitrogen fixers in those samples. And with those few examples of around a few dozens of initial example images, we were able to generate already a machine learning model that was applied to the 2 million images. And this was used to predict new nitrogen fixers images the predictions were curated in order to improve the model and run again. And this was an iterative process until no new images appeared. So the results from the images and then results from the metagenomes were then compared, for example, in terms of biogeography, and they were very consistent, and they allow us to discover new hotspots for nitrogen fixers that were not described in the literature, or at least in certain database that have been collected from these organisms. For example, the Madagascar Channel, some parts of the South Pacific Ocean, some parts of the South Atlantic Ocean near South America, but mainly the Indian Ocean. And all these regions are, let's say, not undersampled. So these were nice results in order to, let's say, convince the scientific community to take more samples from those ocean regions. And with the images, we were able to detect the lifestyles, for example, detect free filaments of trichodesmium, the free filaments of calotrix, as well as filaments of calotrix attached to the diatom ketoceros, filaments of Rickelia living inside the diatom Rhizosolenia or Hemiaulus, colonies of Crocosfera, as well as Crocosfera living inside the diatom Climacodium, as well as the UC9 symbionts that are now considered to be an organelle of certain aptophytes. And as I mentioned, the images allow us to do absolute quantifications, and in terms of absolute number, in terms of cells, the most abundant nitrogen fixers that we detected were those that were not non-photosynthetic. But in terms of biomass, the most abundant one was dominated by trichodesmium. And also the integration between DNA and imaging allow us to deal with certain technical problems. For example, or let's say the intercalibration between the signal from the metagenomes and the imaging. And this intercalibration allow us to quantify polyploidy, the number of genomes that a cell has. This is a problem for quantifying plankton using DNA. And well, we were able to, for example, to inform the higher polyploid level of trichodesmium regarding Rickelia. So there is an overestimation of certain nitrogen fixers towards others when analyzing molecular data. And finally, we were also able to, let's say, suggest new symbiosis. For example, certain metagenomic reads were very similar to the reads from symbionts that were so far detected only in freshwater environments. These symbionts, these metagenomic reads were mainly present in a size fraction where it is considered to be the, the size of the host, and in multiple and distant ocean regions. After that work, another independent scientific group, was able to effectively isolate this symbiosis that seems to be what we saw from the Ocean's metagenomes. And also, we detect a high amount of pennate diatoms containing some granules without chlorophyll autofluorescence, and they were detected mainly in seawater samples that were hotspots of nitrogen fixers, and then very recently was found that they correspond to a new type of symbiosis. So in this part of the work, you can see I wanted to show an example of how this the integration of omics and imaging technology allow us the study of a very important functional groups, as they are nitrogen fixers. And some of the main conclusions were that the, let's say, heterotrophic nitrogen fixers were the more abundant overall in terms of cell abundance, but trichodesmium dominated by biomass. Larger symbionts dominated the tropics, while those free livings tend to be more widespread. We detected new areas that are hotspots for nitrogen fixers, and also we suggest the presence of new symbiosis. And also we suggest the presence of ultra-small nitrogen fixers in the Arctic, which is very interesting to continue studying. But all in all, nitrogen fixers are diverse in all size fraction, globally distributed, but with a patchy biogeography. So another project was more related with other important functional trait or functional group that are phytoplankton. As you know, phytoplankton is study of phytoplankton is very important from different aspects. For example, they are responsible of half of the global photosynthesis of our planet, and they are key players in the biological carbon pap. Um, um, an important issue about phytoplankton is that they are phylogenetically very diverse. They consist of some lineages of cyanobacteria, like Synechococcus, Prochlorococcus, Trichodesmium, Rickelia, as well as mm, many different lineages of eukaryotes that obtain their chloroplast through different events of endosymbiosis. So they cover different, let's say, branches of the tree of life. Um, at that moment, this has can generate some limitation in the traditional ways of doing genetic surveys for so let me explain it a bit better. So molecular surveys of phytoplankton are usually carried out with targeting the genes coding for the ribosomal RNA. This consists of amplifying um, a region of the ribosomal RNA by PCR and then sequencing the amplicons. And this type of approach are, let's say, are very powerful, but at the same time, also they have um, some limitations. For example, the primers that we use and the PCR conditions always introduce biases in which certain taxa are over amplified. Also, there is a problem with the copy number variation of the ribosomal RNA genes, and um, this is not let's say this is not a small problem. To have an idea, um, some chlorophytes has only one copy of this gene, whereas many dinoflagellates have dozens of thousands and thousands of copies. And then other limitation is that these genes are housekeeping, so it's difficult. We cannot use them to distinguish between photosynthetic and non-photosynthetic species. Um, we can do some assumptions in terms of phylogenetic distribution of the photosynthesis capacity, but these assumptions are, let's say, um, a limitation. What I mean is that uh, uh, as I mentioned, the chloroplast has been gained in independent events during evolution in different branches of the eukaryotic tree of life. And in addition, they had been lost multiple times independently in the different lineages. So there are many cases, for example, in dinoflagellates, in which closely related species, uh, in closely related species, we have cases in which one species is photosynthetic in the traditional way, so we can consider them phytoplankton, whereas others are, let's say, heterotrophic. Finally, it's difficult to cover prokaryotes and eukaryotes in a quantitative way because eukaryotes has the 18S ribosomal RNA gene, whereas prokaryotes has the 16S ribosomal RNA gene. Um, this we can also take directly the 16S gene from the chloroplast, but in order to quantify both cyanobacteria and chloroplast, but at this chloroplast ribosomal RNA gene in some cases are very divergent and not distinguished, not detected by these surveys. So yes, the list of limitations, there are many, let's say, points regarding the limitation of these methods. And in this project, the aim was to, let's say, have a picture to develop a new way of analyzing the global phytoplankton community, covering both cyanobacteria and eukaryotes, and in a quantitative way. Um, um, for this, what we the proposal was to directly detect analyzed metagenomes in order to avoid the PCR biases, and instead of analyzing the ribosomal RNA gene, analyzing a new gene. So, and the gene that we end up selecting is the PSBO gene that codified for a subunit of the photosynthesis 2. Um, uh, we selected these genes because it's single copy and is photosynthetic and um, universally present in both cyanobacteria and eukaryotic phytoplankton. And in addition, it has not um, uh, no there is no homologous involved in other metabolism beyond photosynthesis. So when you detect PSBO, you are very sure that the signal comes from a phytoplankton organism. And here, this is the validation. The validation was carried out an analysis by comparing from the Tara Oceans data the different imaging data sets with the 18S DNA, the ribosomal DNA, as well as the PSBO DNA. In this case, you can see the comparison between microscopy and the 18S ribosomal DNA. In the horizontal axis is the ribosomal DNA gene, the ribosomal RNA gene, and in the vertical axis are the quantification based on microscopy. In the first row correspond to the one safe fraction between 5 and 20 microns, and the second row for the larger safe fraction. You can see that in general, this gene, this molecular method, tend to work more or less okay with dinoflagellate and diatoms at some point, but it's but underestimate by a lot other type of eukaryotic phytoplankton like aptophytes and other type of taxa. And this is the same graph that compares microscopy with PSBO. And you can see that there is uh, an improvement in terms of, let's say, correlation, as well as in terms of, let's say, a more closer one-one relation between the microscopy and the DNA counts. This is clear for phytoplankton groups different from diatoms and dinoflagellates, but also there is let's say an improve in the correlation for dinoflagellates and diatoms. Well, these comparisons are only from here from eukaryotic phytoplankton, but then we went to again to the Tara Oceans data to generate a global overview of the complete phytoplankton community, covering eukaryotes and cyanobacteria. And here are I am displaying the biogeography from in the upper map, the smaller size fraction, and the lower map, the larger size fraction, and there are some patterns that make sense. For example, you can see that in the smaller size fraction, the colors correspond to dark blue and light blue that correspond to the Picocyanobacteria Prochlorococcus and Synechococcus that are abundant in tropical and subtropical regions. And in the largest size fractions, there tend to be some yellow pie charts at high latitudes that correspond to diatoms. But one, I mean, in addition to all these patterns that make sense, it was interesting and unexpected to find a high abundance of Synechococcus in the larger size fractions, and also Prochlorococcus of Picocyanobacteria. This is the same data, but in which I display the average across the entire Tara Ocean route for each size fraction. And you can see, again, in the smaller size fractions, a prevalence of Prochlorococcus and Synechococcus. In the larger size fractions, more abundant of eukaryotic phytoplankton, but also some cyanobacteria that we expect to be in the larger size fractions, like the Trichodesmium, or that forms colonies and filaments, and symbionts like Rickelia and Calotrix. But still, the quantity of Prochlorococcus and mainly Synechococcus was unexpected. There can be a technical problem of clocking in the filter, or that so it can be also a technical yes, a technical problem, but still the quantity of this Picocyanobacteria was very abundant. And mm, these are patterns regarding PSBO. So these are patterns that correspond to the relative abundance in terms of cell abundance. When I did some, let's say, biovolume corrections for the different taxa, here I did that, and I plot again the biogeography in the second column of maps. You can see that in terms of biomass, the Picocyanobacteria are very low abundance in the larger size fraction. That makes sense because they are very they are very tiny regarding the other organisms. So once we detected these patterns for PSBO in the metagenomes, we came back to the confocal images to see if we can find this something that support the presence of Picocyanobacteria in the larger size fraction. And we end up detecting a high abundance of these images that correspond to Picocyanobacteria symbionts in dinoflagellates. You can see from the size bar that the size is consistent with the cyanobacteria for Picocyanobacteria. And also, well, I will explain a bit more about this type of images. These images are from confocal microscopy, correspond to different channels, and each channel corresponds to a different proof. Or in the case of the red channel, it corresponds to the autofluorescence of the chlorophyll. So you can see that these little points has chlorophyll, and the size is consistent with the size of Picocyanobacteria. And in addition, we obtain also high amount of colonies or aggregates for Picocyanobacteria. This type of symbiosis or these aggregates has been informed in the literature, or actually also Rachel was one of the first researchers informing this type of symbiosis. The unexpected thing was that was very absolute abundant and widespread. And here you can see again at the at the global level, targeting the symbiosis and the aggregates. You can see that the this is the data, exclusively the data from imaging, nothing about PSBO. You can see the symbiosis are widespread, tend to be widespread and in tropical and subtropical regions. And the aggregates are detected in many areas of the ocean, but especially in certain samples of the South Pacific. Below, there are some other examples of this type of images that were very abundant in the confocal Tara Ocean's confocal microscopy data. So that was one of let's say this work consists of other aspects, but for example, we developed a database from PSBO, and now people are using, there are many independent groups that are already using this reference database from PSBO for analyzing their own metagenome. So I didn't go more deeply with that due to the sake of time, but so I will jump directly to the conclusions in which so for this work we select and validated a new gene for analyzing the entire phytoplankton community. We found that PSBO has improved correlations with microscopy and also flow cytometry than for ribosomal RNA gene. We generated a curated databases of PFU sequences that are now used by the scientific community. We detected high abundance of cyanobacteria aggregates and symbionts in the ocean. All in all, PSBO is a promising new gene marker for molecular-based evaluation of the entire phytoplankton communities. And another way, let's say another example of integration of the data integration, different data set integration, also is related to the analysis of PSBO. So with collaboration with research with Ro El Hurani, who is a researcher specialized in satellite remote sensing, we were able to, let's say, parametricize the satellite data, the optical information that is obtained from satellite data, compare that with the PSBO abundances that we detected in each of the Tara Ocean's sampling stations. And we were able to generate or estimate global biogeography of the different main phytoplankton groups, covering the whole ocean, the whole maps. Um, this is a very promising approaching of integrating omics and satellite for analyzing biodiversity at global scale, because with the satellite data we have the advantage that we we can cover the whole ocean and also in a high temporal resolution. Um, and with PSVO, we are able to incorporate information to the satellite dataset, incorporate information regarding the taxonomy of the organisms at in a very accurate way. One of the main limitations currently for this integrative approach is that still the number of samples for um, the the number of metagenomes obtained from the ocean still is not so big. We are talking about 100 or 120 sampling stations. Um, Currently, Tara Oceans is taking new samples in new ocean regions, and also there are some other stations, for example, in the Pacific, and new sampling station in the Atlantic, in which they have been already sampled, and the 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 consortium is in the process of analyzing the DNA data. So with time, we will imp there will be an improvement of the coverage of the ocean, and this approach of integrating the omics and data and satellite data can be very useful for biodiversity assessment. Finally, this is a very recent project um, that we finished very well. We finished these days, and this is focused more in a specific group of phytoplankton, the diatoms. As you know, they are and they are many very important to study due to different characteristics or different implications. For example, they are responsible of around 20% of the global photosynthesis of our planet. Also, they are covered by a silica cell wall, so they are very important not only in the carbon cycle but also in the silica cycle. As I mentioned in the previously, they make association with nitrogen fixers, so they are also important in the nitrogen cycle. Um, in this project, the main questions that we decided to tackle were what are the global biogeography of diatoms for the different diatom taxonomic groups and the different genera. Which regions are hotspots of diversity and abundance? What are the environmental factors that drive their distribution and their community structure? And how they adapt and acclimate to the different environmental conditions. So in this case, this project integrated the analysis of the 18th ribosomal RNA gene because it allows us to go very deeply in the taxonomy and detect diatom genera that are very low abundant, and also integrate data from optical microscopy because diatoms are very, due to their morphological, different morphologies and shapes, they are easy to annotate in relation when compared to other phytoplankton groups. And finally, also we were not only interested in terms of taxonomic diversity of diatom, but also in terms of functional diversity of diatom. So we also analyzed for this project the metatranscriptomes. I mean the RNA sequencing data sets. Um, and well, and there is another technical thing that in the analysis of the ribosomal RNA gene, we compare uh, two different regions of the gene. One that is called B4 and another that is called B9. In general, the um, the 18S ribosomal RNA gene is not analyzed the whole gene. Nowadays, with the new technology, this is started to be done, but in general, the gene is analyzed for certain regions that are surrounded by conserved uh, areas that are enough for designing primers, and the regions in itself that are amplified are different enough to be able to distinguish different species and genera. And also the length of the region should be according to the i- short enough to be able to be sequenced with the most popular sequencing technology. For example, the B9 region is around 130 pair bases, and the B4 region is around 385 pair bases. Um, so in this, so to summarize, in this case, um, we wanted to compare the two different 18S regions, B4 and B9, with optical microscopy, and finally to go beyond taxonomic diversity and by covering also functional diversity with the analysis of RNA sequences. Um, well, for the sake of time, pass. Uh, I will go very quickly with the analysis of the biodiversity of diatoms. Um, I can mention that we detected the most abundant genera in the ocean. The result, when compare the analysis of the B4 and the B9 uh, genes were very consistent. And in addition, they were consistent with the microscopy data. And okay, so we were able to detect the main diatom genera. Some of them were cosmopolitan, but more abundant in the Arctic and Southern Ocean, as in the cases of Chetoceros, Thalassiocyra, Actinocyclus, and Fragilariopsis. Others are more cosmopolitan, are also cosmopolitan, but more widespread distributed, or let's say more homogeneously distributed in the different ocean areas, as in the case of Pseudonesia. Other genera like Porosira and Proboscia were detected exclusively at high latitudes in the Arctic and Southern Ocean. Others like Fragillaria were exclusively detected in the Arctic, and whereas Coretron, Trieres, and Hemiaulus exclusively detected or mainly detected in the Southern Ocean. And finally, some genera like Guinardia and Leptocylindrus were prevalent in tropical and subtropical regions. We were able to define uh, different diatom communities that were covariant, uh, and these communities has vary also in their environmental preferences. I will not go deeply with this, but I can say that the main important point here is that in the last, I mean, in the bottom panel, it shows the ocean distribution of the different communities. The communities are organized by the dendrogram in the upper part according to their similarities in terms of environmental distribution, and you can see then that in the lower panel how they distribute in terms of ocean regions. One important message here is that the Arctic Ocean that is in red, the is displayed in red. The Arctic Ocean stands up as a diatom hotspot because 6 of the 25 communities were exclusively or mainly detected in that area. And we also detected a high amount of amplicons of sequences that we have no clue regarding the species of the genera. These unassigned sequences are due to the limited in our reference databases. Is we have to take into account always that environmental samples are all, in terms of DNA analysis, are always analyzed in comparison with reference databases, and sometimes there are limitations. For example, the most here the most abundant unassigned sequences came from the Southern Ocean. Here you can see in the lower panel. So in the Southern Ocean, most of the unassigned sequences either from B4 or from B9 region. In both cases, they came from the Southern Ocean. So this, let's say, illustrate that there are still many parts of the ocean that are undersampled, that we need to improve their coverage in terms of cultural isolation, as well as in terms of environmental sample analysis, in order to improve our reference databases. But well, I went very quickly from the part of the analysis of taxonomic diversity, but because I want to go and mention a bit about the analysis of the functional diversity in diatoms. So as I mentioned, we analyzed all the RNA sequences datasets from the whole Tara Ocean transect from diatoms. And here I am showing the top 100 most abundant transcript families. In the upper panel, how is their abundant distribution, and in the lower panel, how is their ocean distribution. One important message is that the most abundant transcript family, correspond to the LHCs. LHCs correspond to the light harvesting complex, or chlorophyll A-B binding proteins, also known as chlorophyll A-B binding proteins. They are maybe the most abundant, they are considered sometimes the most abundant membrane protein on Earth. And in the case of diatoms, at the global ocean scale, they account for around 3% of the metatranscriptomic abundance. This group of these LHC families contain different subfamilies, and the different subfamilies can, some subfamilies are involved in the light harvesting, whereas other subfamilies are involved in the opposite activity, in the photoprotection. I mean, in the dissipation of the excess of light. And this is, so the ratio in expression in the different subfamilies is very important in order to fine-tune photosynthesis, because photosynthesis is consists of the electron transport of, let's say, in the photosynthetic chain, and an excess of electrons can generate damage to the cell, and a deficient of electron transport can generate lower growth. So it's very super important, the fine tuning of between light harvesting and photoprotection. Um, so in this here we analyze the ratios, the abundance of in the or the expression of the different subfamilies in the surface at around 5 meters in relation to the deep chlorophyll maximum that was around 1,000 meters, but varies according to the ocean area. Um, uh, we detected that photoprotecting subfamilies were prevalent at surface, whereas those subfamilies involving light harvesting were predominant in the deep chlorophyll maximum. And we also detected patterns regarding the size fraction. In particular, there is a higher light harvesting um, light harvesting expression, or in larger size fractions, whereas the opposite is regarding photoprotection. And we think that these patterns are related to, or are associated, um, or are explained in order to counteract the intracellular light attenuation. Um, because when the cell volume increases, the length path of light within the cell increases, and this reduces the light intensity with per unit of volume. Um, so this is some examples of how the photosynthesis machine is fine-tuned according to conditions like depth or cell volume. Finally, another interesting issue regarding this analysis is that among the top 100 most abundant transcript families, uh, we only detect one that is unknown. Most of the rest of the families, we can have some insights regarding their function, but there is one family that is called DAF 285, and DAF is the acronym of Domain of Unknown Function. And when we analyze the environmental distribution of this, the expression of this family, they are highly expressed at high latitudes in the Arctic and the Southern Ocean. And nowadays, with the new methods that there are new methods like AlphaFold that we can predict with high accuracy the structure of a protein using exclusively the primary sequence of amino acids. So and for these subfamilies, these are the shapes of the predicted shapes for the protein. And these proteins are what is called solenoid conformation, in which there is a repetition pattern of a secondary structure. And these type of solenoid proteins have been detected in proteins with antifreeze activity. So the both issues, the protein conformation and the predicted protein conformation and the prevalence in high latitudes, allow us to raise the hypothesis that this protein can be related to the as an adaptation to low temperatures. And this is a nice example of how environmental data and the integration, different aspects of integration, including of environmental data, including the environmental distribution, but as well as 3D structure can guide us to generate hypotheses that eventually this can be evaluated in the lab. Um, so in this project, um, I went very quickly, but basically the project was about in this project we were able to detect the most abundant diatom genera at global scale. And there was a methodological agreement when comparing uh, B4 and B9 regions of the ribosomal RNA gene, as well as light microscopy. The most diatom was the most abundant eukaryotic photosynthesis group in these datasets, with prevalent genera like Ketocero, Thalassiosira, Actinocyclus, and Pseudonychia. In addition, we were able to define different diatom community with varying environmental preference, and 6 of these communities were mainly present in the Arctic, and this illustrates the Arctic Ocean as a hotspot of diatom diversity. And in terms of functional insights, we detected gene, uh, uh, we detected expression patterns for the LHC family that are related to photosynthesis acclimation according to the depth partition, as well as according to the cell size. And we detected the high abundant expression of the enigmatic family DAF 285 that was very expressed and high latitudes. And these were basically three examples that, as I mentioned, are examples of analyzing specific phytoplankton groups by integration of different and massive data sets, including different type of omics that can be metabarcoding of a certain gene like the 18S ribosomal RNA gene, or metagenomic analysis of marker genes like PSBO of NIFH, as well as and also molecular data related to functional analysis like metatranscriptomics, and also high throughput imaging data. Um, um, and finally, I want to highlight that in the last case, in this case, in which we can raise a hypothesis analyzing for an unknown family, analyzing the 3D conformation of the protein as well as the environmental conformation. This is an important issue because when we detect the genes of a given seawater sample, when we analyze, let's say, the metagenome from a given seawater sample, most of the genes or half of the genes tend to be unknown, exclusively unknown. This is, for example, a recent paper in which they analyzed the whole a whole public database, and they analyzed for the metagenome that were there, and you can see that marine samples were one of the samples that contain most of the unknown genes that we have collected so far. So there is still a huge, let's say, field to target regarding what these unknown gene families are doing. And now with the new technologies that analyze proteins or predict protein as 3D structures, or are able to analyze proteins in a very complex way, we can go more deeply in this analysis. So, thank you. Part of this analysis were done in my previous postdoc in the lab of Chris Bowler, and also including one project in collaboration with Rachel, and more the more recent project regarding diatom has been done also in collaborating with the lab of Chris, and also with my current uh, during my current postdoc in the lab of Sergei Ovkinokov. And well, I am open to questions, and thank you.